Central Dogma. Table of Contents. Central Dogma, Transcription, mRNA processing, the genetic code, translation. Central Dogma. From DNA to protein. Proteins are the machinery in charge of carrying out important cellular functions, such as the synthesis of glucose via photosynthesis or the creation of ATP via cellular respiration. But how does the cell know which protein to make? And how is the cell able to consistently make the correct protein that it needs? In this unit, we are going to study DNA more in depth. We will see how its structure makes it a suitable molecule for information storage. We will also explore the mechanisms that the cell uses to access the instructions in DNA in order to build proteins. As cells multiply, DNA needs to be copied so that the daughter cells can also acquire the instructions to make proteins. This process is called DNA replication, indicated by the arrow on the far left. The process of replication, transcription, and translation are so critical that they are called the central dogma of biology. Transcription occurs in the process of DNA going to RNA, and translation is the process that occurs when RNA is translated into proteins. With few exceptions, the process only flows from DNA to RNA to protein. A dogma is a postulate, an idea, a philosophy. It is central because it is what life is based on. The transfer of information from genes to proteins is usually a one-way process. Changes in DNA affect RNA and protein, but changes in proteins or RNA do not often affect DNA. This will have an important implications when we study genetics. The exception to this rule is that some viruses copy their RNA to make DNA when infecting a cell, which can have impacts on infected individuals and on evolution. The segment of DNA needed to make a specific protein is called a gene. Gene expression is the process of taking the information from the gene and making the protein it codes for. The central dogma of biology is basically an explanation of how gene expression takes place. This process occurs whenever a specific protein is needed by the cell. So what we see in the image is the primary structure of a protein. It's a sequence of amino acids. Here are the steps of gene expression. There are two. First, the gene is copied from DNA into RNA through a process known as transcription. Then RNA builds the protein through a process called translation. Gene expression in prokaryotes versus eukaryotes. Transcription and translation occur both in eukaryotes and prokaryotes. However, there are key differences. One, prokaryotes do not have a nucleus. Their DNA is free-floating in the cytoplasm where both transcription and translation occur. Translation starts before transcription is even finished because the process happens all in the cytoplasm. In eukaryotes, though, the nucleus provides a physical barrier that separates the two processes. Transcription only occurs in the nucleus. It is finished, and then translation is started. So in the prokaryotic cell, gene expression looks like this. We have DNA. Transcription is occurring where the DNA is being transcribed to RNA. or mRNA, and simultaneously that mRNA is being translated into a protein or a polypeptide as it's labeled here. So transcription and translation occur simultaneously in the cytoplasm of a prokaryotic cell. In the eukaryotic cell, transcription takes place inside the nucleus. So we can see our DNA here, transcription of DNA into RNA happens. The RNA molecule that results from transcription is not ready for translation. Okay, pre-mRNA as it's called, it's not ready for translation. It has to undergo RNA processing first. After processing, the RNA is known as mRNA, messenger RNA, and that moves to the cytoplasm where the translation occurs. Transcription and translation steps. Transcription and translation both have 3 steps called initiation, the beginning, elongation, where the RNA in transcription or the protein in translation is made longer, and termination, which is the end. The activities that occur at each step are different for transcription and translation, but they have the same names. Regulation of gene expression. How does a cell know what genes to express or what proteins to make? Are all proteins made at the same time? Prokaryotes and eukaryotes have different ways to regulate gene expression, or in other words, turn genes on or off depending on the cell's needs. Prokaryotes regulate the expression of genes by controlling transcription. Eukaryotes, on the other hand, can control gene expression at many points. Gene expression regulation will be discussed in more detail in the next unit. A chromosome is an organized structure of DNA and protein found in cells. It is a single piece of coiled DNA containing many genes. Chromosomes vary widely between different organisms. The DNA molecule may be circular or linear and can be composed of 100,000 to over 3,750,000,000 nucleotides in a long chain. Typically, eukaryotic cells have large linear chromosomes, and prokaryotic cells have smaller circular chromosomes. Prokaryotic chromosomes. This is a chromosomal map of a bacteria, H. orenii. Like all bacteria, this circular DNA molecule contains all the genes that are needed to make the entire organism. This particular bacterial genome is made up of approximately 2,500,000 nucleotides. Each different color in the outer circle represents another gene. Eukaryotic chromosomes. In eukaryotes, nuclear chromosomes are packaged by proteins into a condensed structure called chromatin. This allows the very long DNA molecules to fit into the cell nucleus. Chromosomes are the essential unit for cellular division and must be replicated, divided, and passed successfully to their daughter cells to ensure the genetic diversity and survival of offspring. Since eukaryotes are larger and more complex, eukaryotic chromosomes are much larger and require more complex methods for storage of their numerous genes. Special proteins called histones help pack the DNA strand into tight coils. Additional proteins called scaffolds help maintain chromosome integrity. To keep DNA organized in the nucleus, the chromatin of leukocytic DNA is wrapped around histone proteins. This forms subunits called nucleosomes, which in turn form coils, and then it is coiled again into supercoils and ultimately a chromosome.